Hello, and welcome to this Biology 206 Human Physiology Lab overview for the Salivary Cortisol Levels Laboratory. So as always, a few objectives associated with this lab. These are also copied over to the lab report template so that you have these available. And keep in mind that these, as well as the questions at the end of the lab report, as well as in the lab study guide, are going to help outline those types of things that we feel are going to be very important for you to know from these lab activities, and our possibilities to see on the lab exam coming up in a few weeks. So to start out with, this week's lab, we're going to be taking a look at cortisol levels within the body. And so what we want to do is start out with this kind of very basic understanding about what cortisol is. And so cortisol is going to be an example of a steroid hormone. So as a steroid hormone, it means basically it's built around a cholesterol ring. So in this case, we've got cortisol over here on the left, aldosterone, another steroid hormone, as well as an androgen over here, a dihydroxyepiandrosterone. And so you can see they all have the basic cholesterol ring structure, which is going to be present, but then we have alterations in the functional groups that are going to be there. And so if we take a look at this between cortisol and aldosterone, we can see that in this case we've got an HCO that is present in an aldosterone and it's missing here, and in place what we've got is a hydroxyl group located here where we don't have a hydroxyl group over there. This one here again has different modifications to be present. Now cortisol is going to be a hormone that is secreted by the adrenal cortex. So the adrenal gland, adrenal basically means on top of the kidneys, and so they've got the kidneys located retroperitoneally within the abdominal cavity, basically on the back wall, kind of on either side of the aorta and the vena cava. The adrenal gland sitting up here almost as a little cap. If we take a look at this kind of through the section of the adrenal gland, we're going to see a cortex around the outside, a deeper medulla region right in there. And so if we take a look at this, again, as we were to look at it underneath the microscope, we can see different regions within the adrenal cortex, deeper regions down here in blue for the adrenal medulla. And it's going to be cells within this middle region, the zona fasciculata, that are going to be involved with secreting the cortisol. And so if we take a look at it, cortisol is going to be a hormone that is involved with a whole variety of processes within the body, but we can see that it is also under circadian rhythm control. So that in general, for people that sleep at night and are active during the day, what we're going to see is that their cortisol levels are going to be naturally lower overnight, and they're going to start to increase as an individual starts to wake up, and they're going to be high throughout the day. And so that is showing a circadian rhythm control. This is showing a difference between high levels of cortisol during the day, low levels of cortisol at night. And so if we take a look at this, basically cortisol is described as a stress hormone. This is a hormone that is going to be released by the endocrine system, basically released by the brain through the anterior pituitary in response to different types of stress. And it may be physical stress associated with exertion, emotional stress, anxiety, stress, things like that affecting individuals, as well as metabolic changes. And so what we can see in this slide, again, indicating the control mechanism for the release of cortisol, we've got the hypothalamus kind of setting up here. And then cells within the hypothalamus are basically going to be sensing what goes on within the body. And so, like I said, it could be physical stress, emotional stress, metabolic changes like hypoglycemia, as well as that circadian rhythm kind of going in and contributing to that. So basically, we're going to have cells in here within the hypothalamus that are going to be involved, hopefully my next slide is correct, with releasing what's referred to as corticotropin releasing hormone. And so we're basically going to have cells located within the hypothalamus responding to these kind of stress stimuli. And they're going to basically recognize those stress stimuli, release this hormone, cortical releasing hormone CRH, which is going to get into the circulatory system, get into a series of blood capillaries, be transported through this kind of relatively small kind of distance here into the anterior pituitary, where that corticotropin releasing hormone CRH is going to stimulate cells here within the anterior pituitary to secrete ACTH. ACTH is adrenocorticotropic releasing hormone. Now, it's not all of the cells. It's going to be cells that are specifically responding to corticotropin releasing hormones, to CRH. So they have receptors that are located within their cells. The ACTH then is going to be released, ultimately going down to the adrenal gland, where it's going to affect cells within the adrenal cortex to release cortisol. Now, this control mechanism, like many of the other control mechanisms within the body, is going to be under a negative feedback system. And so we can have a short negative feedback loop and a long negative feedback loop. The short negative feedback loop is basically the ACTH, the adrenocorticotropic releasing hormone, released by the anterior pituitary in response to the CRH, the corticotropin releasing hormone. That ACTH can filter back, feedback onto the hypothalamus and say, okay, we got the message. We're sending the ACTH out to the body, and so this can kind of limit the production of CRH. Now, that's the short feedback loop going from the anterior pituitary to the hypothalamus as a result of the ACTH, the signaling hormone. Now, the ACTH is also going to go down to the adrenal gland, where it's going to stimulate the release of cortisol. Now, that cortisol is going to be involved in a long negative feedback loop. That long negative feedback loop is going to go back to both cells within the anterior pituitary as well as cells up here within the hypothalamus. And so the ACTH feedback, the short feedback loop, is basically saying, yeah, we got the message is being passed on. The cortisol feedback loop over here is saying, yes, we are producing this stress hormone. We are producing what we were told to produce. And again, with negative feedback, this is going to feed back onto the cells within either the anterior pituitary or the hypothalamus, so that they are, controlling the amount of cortisol that is being produced. We don't want to produce lots and lots of it. We want to maintain kind of the appropriate level of that within the body. We don't want to waste resources. Now, the actions of the cortisol, again, as a steroid hormone, it needs to be carried by a corticosteroid binding protein. So basically, a steroid hormone built on a cholesterol ring is going to be fairly hydrophobic. It's not going to be able to get into this aqueous environment within the circulatory system really easily. And so this corticosteroid binding protein is going to bind to it, take it out to the periphery, and then allow this to get into cells that are going to need to respond to it. So cortisol is a steroid hormone, be able to cross across the cell membrane, get into the intracellular region, into the cytosol, where it's going to bind to intracellular, inside of the cell receptors. And basically, it is going to act as a transcription factor. The interaction between cortisol, the steroid, and these receptors is going to activate or control what's going on within the DNA, what genes are going to be expressed. Now, the overall effects of cortisol are things like reducing inflammatory response, increasing gluconeogenesis, making energy available, potentially breakdown of muscle proteins. It's involved in calcium homeostasis, and a variety of other functions, some of which that we're going to cover in lecture within the course. So that's the cortisol. But now we need to be able to measure it. And the way that we're going to measure cortisol within the laboratory this week is to do an enzyme-linked immunoassay, an ELISA. And so the concept with an enzyme-linked immunoassay is that we're going to use antibodies. Antibodies over here are these little Y-shaped proteins. And as we're going to talk about when we get to the immune system, these antibodies are going to be very specific at binding to their target. In this case, the antibodies are going to be very specific at binding to the little red dots, and the little red dots are going to represent cortisol molecules. And so we're going to put the antibodies into a plate, and so we got a multi-well plate kind of located down here. Each one of these little wells is going to have antibodies in it, and we put different samples and run through this different reaction. And so we're going to take our antibodies located on our plate, mix it with our sample, and basically if cortisol, or if the little red dots are present within our sample, they are going to be able to bind to the antibody and be trapped there. We're going to wash it off, use a secondary antibody, so another antibody is going to be added. In this case, this is an antibody that has some type of marker associated with it. The example we've got here is a marker that is going to cause an enzymatic reaction and a colored precipitate forming. And so we see the color change, and it'll basically kind of drop out of solution, and we'll see a color change within this well. And then once we've done that, the amount of cortisol and the amount of red dots that are present there is going to be related to the amount of colored precipitate that we're going to see. We can put that into a plate reader, and it'll go through and measure the amount of color that is going to be present. So that's the basic process for an enzyme-linked immunoassay. What we're going to be doing is a competitive ELISA, a competitive enzyme-linked immunoassay. And so it's going to be competitive because basically we're going to have whatever it is that we're going to be analyzing. In this case, this diagram, are analyzing our cortisol for our samples, are going to be little green dots, and it's going to be our sample cortisol coming from the saliva from our classmates, is going to be competing for binding sites on these Y-shaped antibodies with this labeled antigen. And so these little gray dots with the little red stars on them are going to be cortisol that is provided in the kit for doing this competitive ELISA, this competitive enzyme-linked immunoassay. And so what will happen, we'll put in a known amount of our labeled antigen, a known amount of cortisol molecules, gray molecules with little red stars on them, indicating that we can measure kind of the color that's going to be there. They're going to compete for binding sites within these antibodies over here. So, yeah, that's nice, but how does that actually, how does that actually work? And so we take a look at this. On the top situation here, we're going to see a sample that has relatively high levels of our analyte, the relatively high levels of these green dots, and relatively low levels of our labeled antigen. And so again, the gray is what comes in the kit. This is cortisol with the colored marker already placed on it. The green are going to be the cortisol that is at higher concentration in the saliva of one of our classmates. And so a competition, if we've got more green dots than gray dots, we're likely to have more green dots attached to our antibodies than gray dots. So in this case, we're going to have more of the unlabeled cortisol, the sample cortisol, than we are our labeled antigen, our labeled cortisol coming from the kit. In contrast, if we have a sample with a low level of cortisol within it, sample with low levels of cortisol from our saliva from our classmates, we're going to have a relatively higher concentration of these gray dots. We're going to have a relatively higher concentration of the cortisol that came in the kit and is labeled with the marker. And so if we load in the same amount of labeled antigen, the gray dots with the red stars, with the marker being present, we can, based on the competition between the green dots and the gray dots, between our sample cortisol and our labeled cortisol from the kit, we can do a measurement, and you know, do the appropriate kind of controls. You can look at the level of the antigen and plot it out and be able to determine how much cortisol was present within the saliva. So that's the competitive immunolysa, competitive enzyme-linked immunoassay. Now, it's a process basically moving a lot of samples back and forth and rinsing them and adding chemicals that we're not going to do in lab. I'm going to do that outside of the lab and provide the lab results for you. So this week in lab, what it's actually going to be doing, again, like all of the labs, you're going to need to download the template, lab report number 2. It's available on a link. It says template. This is a Google Doc. You're going to need to download this to your computer or copy it to your Google Drive, and then edit that, and then ultimately upload it later once you've completed the activities and answered the question. Now, previously, we had our volunteer students spit into a test tube. We used Eppendorf test tube, 1.5 millimeter Eppendorfs instead of these, basically collecting samples 30 minutes after waking in the morning and between 9 and 11 p.m. In the evening. Those samples were frozen and then made available, and then we ran the enzyme-linked immunoassay. And so what you're going to get are going to be the results of these immunoassay from the samples. Activity number 1 is going to take you through descriptive statistics and inferential statistics, basically saying, are these cortisol levels higher during the day than they are at night with our classmates. Activity number 2 is adding in a little bit additional kind of scientific experimental evidence looking at salivary cortisol levels in relationship to physical exertion. So the first activity is going to be taking a look at exercise effects on salivary cortisol levels at about 40% VO2 max. 40% VO2 max is, kind of limited, kind of somewhat strong or somewhat exertion, some kind of basic level of exertion. Fairly light, not overly exertive. And so basically we're going to take a look at that again with descriptive and inferential statistics. Then we're going to do activity 2B, where in the previous one, where we did 40% VO2, which is light intensity, at 80% VO2, we're going to be taking a look at a very hard intensity, a very hard individual kind of exertion scale, exertion levels. Again, taking a look at this, doing descriptive statistics and inferential statistics, taking a look at basically between the 40% VO2 max and the 80% VO2 max, looking at the effects of exertion, physical stress, on these cortisol levels. Once you've completed those activities, the questions at the end of the lab report, you're going to submit your lab report through Canvas by uploading either the Google Doc link or your Microsoft Word document. As always, your individual laboratory instructor may have further details and updates available to you. Please let me know if you have any questions or concerns. You can either let me know or your laboratory instructor. Recognize that we are here to support you throughout this semester. Thank you and have a great day.